Hi guys, today we are going to study a process which is called meiosis or gametogenesis. The gametogenesis is the formation of gametes. Gametes are ovumes and spermatozoons. So, for understanding this, is absolutely necessary you have understood the mitosis. If you don't understand now mitosis, you should revise and study the mitosis, and then you will understand meiosis. Before studying this, we are going to see this. As you know, human cells are made of 23 pairs of chromosomes. These chromosomes are the DNA, which is coiled, okay, and contains the genes. So, for every single gene, we have two different versions. One comes from our father, and the other one comes from our mother. One from our father, one from our mother. For example, and these ones are different. They have the same, every single pair of chromosomes have the same genes, but not the same information. I mean, in this chromosome, I can find the gene for the color of the eyes, but the information can be different. My mother gave me the gene of their eyes, which is blue, and my father gave me the gene of his eyes, which is brown. The gene is the same, color of the eyes, but the information is different, blue or brown, okay? So they carry the same information, but the same genes, okay, with different information. So, as you know, we have 23 pairs. So, N is going to be 23. But we are 2N, okay? Which means that we are diploid. We are diploid is that we are 2N. So, N is 23, so we have 23 pairs of chromosomes. 23 pairs, 2N. Which means 46 chromosomes, obviously. Okay. When we produce gametes, ovumes, and spermatozoons, those gametes are going to have the half of chromosomes. The cells that are specialized in producing gametes are going to divide in a process which is called meiosis, and they are going to halve the number of chromosomes. So, ovumes and spermatozoons are going to be haploid cells. They are going to be N, not 2N. So, they are going to have 23 chromosomes, not 23 pairs. After this, fertilization occurs, and when the spermatozoon fertilizes the ovum, it will form a zygote. The zygote is a cell which is diploid, is 2N, because it has 23 chromosomes from the spermatozoid and 23 chromosomes from the ovum. 46 or 23 pairs. This zygote is going to grow. It's going to divide. It's going to divide and form more and more cells. These cells are going to be genetically equal to this one, to the first one. This is called the embryo and is diploid. The embryo is going to continue dividing. The cells are going to continue dividing in order to form a new individual, which is going to be diploid as the parents. The process when this cell divides to form new cells, the cell growth, is the mitosis, because one cell is going to form another cell which is genetically identical. Here I have written stem cells. In Spanish are called células madre. Stem cells are an indifferentiated cell which can form any other kind of cell. The stem cells are in the zygote and in the embryo because they are going to form every single kind of cells of our body. Okay? Now, if you have understood this, we are going to see this process, the meiosis, the formation of gametes. Meiosis has two main parts. The first division, which is here, and the second division, which is here. Second division is just like mitosis, okay? It's just like mitosis. But first division is different. It has the same phases: prophase, metaphase, anaphase, telophase, and cytokinesis, okay? Here we have the prophase 1, metaphase 1, anaphase 1, and here the two: prophase 2, metaphase 2, anaphase 2, okay? So let's start and see what happens. In the first division, we have an oocyte or spermatocyte, which is a cell that is going to form ovumes or spermatozoon, which is diploid, okay? It's a cell of an adult animal or plant. And this cell is going to suffer the first division. In the prophase 1, what happens? That the nuclear envelope disappears and the DNA coiled and form the chromosomes. This is just like in mitosis, but there's a difference. In the first division of meiosis, the homologous chromosomes, the homologous are the chromosomes of each pair, okay? The chromosomes of each pair are called homologous chromosomes. They are going to do a process which is called crossing over. Crossing over is to interchange fragments of DNA. They are going to interchange genes, okay? Remember that they have the same gene, okay, with different information, and they are going to interchange it. So the cell that is going to result from here is going to be two cells, different cells. This during the prophase, the nuclear envelope disappear, the DNA coiled forming chromosomes, and the chromosomes do the crossing over. In metaphase 1, the homologous chromosomes, okay, each pair of chromosomes is going to migrate to the equator of the cell. Here, there's another difference with mitosis. In mitosis, the chromosomes went to the equator of the cell, but in meiosis, the homologous chromosomes are the ones that are going to locate here, the pairs of chromosomes. And during anaphase, the mitotic spindle is going to take the chromosomes to each pole of the cell. Another difference with mitosis. In mitosis, the mitotic spindle separates the chromatids of chromosomes, but here the spindle separates the homologous chromosomes. So each chromosome is going to migrate to each pole of the cell. As a result of this, we are going to obtain two cells with 3 chromosomes and 3 chromosomes. This is a haploid cells. Haploid, because we start with 3 pairs of chromosomes, and we obtain 3 chromosomes. Okay? Can you see the difference? In mitosis, we obtain in this part 3 chromatids. But these chromatids are just the same. You know that one is the copy of the other one. Now, during the second division, it's going to happen just a normal mitosis. In prophase, the nuclear envelope disappears. The chromosomes, not the pairs, migrate to the equator of the cell. In anaphase, the mitotic spindle separates the chromatids, and we obtain 2 cells with 3 chromatids. From 3 chromosomes, we obtain 3 chromatids in each cell, but these chromatids are copy one of another. So, here we obtain 4 cells, 4 haploid cells. Differences between meiosis and mitosis. Mitosis, first difference. Mitosis is for cell proliferation, for cell reproduction, okay? For tissue growth, or repairing damaged tissues. But meiosis is for the meiosis is for the purpose of meiosis is making sexual cells or gametes. Okay. Second difference: in mitosis, from one diploid cell, we obtain 2 diploid cells, and in meiosis, from one diploid cell, we obtain 4 haploid cells. Third difference: during mitosis, there's no crossing over, and during meiosis, there's a crossing over. And the last difference, the fourth difference, is that during mitosis, we obtain from one cell 2 identical cells. But in meiosis, from one cell, we obtain 4 different, genetically different cells. These cells are going to form the ovumes and spermatozoons. And lastly, the key of the question, okay, is this process, these 3, okay. This is which makes the difference. These 3 are the ones that make the difference with mitosis. The crossing over, the fact that the chromosomes are in pairs, and the fact, here is the key, here is the fact that during meiosis, the ones that separates are the homologous chromosomes. And in mitosis, the ones that separates are the chromatids, like here, okay? When we saw mitosis, during anaphase, the ones that separates are the chromatids, and here in the anaphase, the ones that separate are the homologous chromosomes. This one is the one that makes the difference, because with this process we are going to obtain 3 and 3 chromosomes instead of pairs, 3 pairs. And so that's all. I hope you have understand everything. In case of any doubt, you can ask, and luck for the exam. Bye.